Hi, my name is Elisa. I am back, and we are talking about the male reproductive system. And this time, we're going to talk a little bit more specifically about sperm and semen. Okay, so we're going to talk a little bit about meiosis, which I hope you went over a little bit before, especially with your biology class. So, meiosis, mitosis. We'll look at the cell types and spermatogenesis, which is the origin of sperm, right? During those stages, we'll describe what the nurse cells are doing during that time, how they're supporting those nascent sperms, developing sperms. Hopefully, what you can do is you could draw your own and possibly label that same sperm cell on your own, and you can describe what all the components are of semen, as well as what they do, how are they important. Spermatogenesis is the process of sperm production in the seminiferous nursery tubules of the testes. It involves three things that gotta happen. Number one, cells gotta divide. Number two, they've gotta divide and remodel larger sperm cells that are actually the germ cells, right? So they're the precursor cells into like smaller, more like aerodynamic and packed up, ready to go sperm cells with the flagella. Number two, we've got to reduce that chromosome number by 1/2, right? Because we are sexually reproducing animals and we have to keep that genome at a stable number. Shuffling of the genes, so each chromosome contains a new gene combination that did not exist previously in the parent. And so that's super important because it gives all of our offspring variation, right? And so that's why all your kids look a little bit different, right? And it keeps that variety. And so we can have more successful outcomes, right, in our offspring. Genetic variation, super important. Meiosis is going to allow for the recombination of genes during one of the steps, which we'll look at, and it also cuts that chromosome number down in half, which is super important. And we're going to result in 4 daughter cells, which are going to be the formed sperm cells that are going to result from that process. So if you could take a look at this, I just would really, if this makes your heart skip a beat from scaredness and sadness, I seriously recommend just getting a couple of these YouTube videos and reviewing this process because it's pretty important. Basically, in a nutshell, what ends up happening is this. Um, in the first stage of cell division, we are going to have what we call recombination. So this recombination is important because it allows for the mixing up of genes. As you can see here, these chromosomes now have mixed components, right? And they kind of follow all the way through. So we splice them out down, and so now we have like genetically different cells from the parent because we've undergone that recombination. Recomb. Yay. The next thing we need to do is we need to cut that chromosome number down in half, right? Because we got to fuse to an egg cell later on. Can't have too many chromosomes, can have disastrous effects if we have like an entire complement. So what ends up happening is this process where we're going to actually split that genome down again in another a round of cell division, where we get haploid cells, right? Haploid. So they have like 1/2 of the genetic material that we need. And they're all single-stranded chromosomes, so they are ready to recombine and they are ready to develop into sperm cells. Hold on, there it is. So, 2 forms of cell division. We just looked at what we call meiosis. I made some gametes, right? Gametes. Got a me in there? Okay. Mitosis we don't use to make sperm and egg cells because that's just really making clones. Like for the most part, we're just doing things like making repairing cells. Cancer also uses mitosis, but we start off with this type of cell division originally. Meiosis is where we need to get to if we're going to make any of those gametes. So by combining a haploid male and a haploid female gamete, we can produce one embryo with all the full set of proper complement of chromosomes to develop into a healthy and happy new human being. Um, boop, there goes. Meiosis has 2 cell divisions. We looked at that. So if you're in the first stage, you're looking at these little ones through everything. Please go back, review all that biology. Prophase, metaphase, anaphase. So that was prometaphase now too. Sorry, forgot about that one. It's new since I went to school. Anaphase and telophase. Before this process begins, DNA is doubled. Prophase is a pair of homologous chromosomes. They're going to line up so that they can do that crossing over thing that is going to make the new combinations of genes, which is going to be great for variations in kids. The problem is each is still double-stranded after, which is why we got to go to around meiosis 2. There's also a prophase, there's also prometaphase, sorry, metaphase, anaphase, and telophase. And all we're doing here is we're just like getting it down to single-stranded, right? Chromosomes, and so we end up with 4 cells because of 2 rounds of cell division. All right. Sperm production, as you might remember, happens in the nursery known as the seminiferous tubules. Here's a cross-section of that seminiferous tubule. And here's kind of what it looks like here. So this is like the lumen, that's empty space. That's like the highway that they would travel down to get to like epididymis, right? And so what we have here is we have like these like spermatogonium, and that's where we start off. And these spermatogonium are going to develop into like a later stage, also known as a type B. Once they've developed far enough, they're still 2N, we call primary spermatocyte. The problem is they have too many chromosomes. We've got to divide it by half. So meiosis 2 occurs here at the secondary spermatocyte, right? So all this is like crossover, and then all of this is like haploiding, right? That's what that's doing. So we're splitting these down until we have a spermatid. Spermatid doesn't have any of the bells and whistles. It doesn't have a flagella. It doesn't have like a component that has all of the enzymes. It doesn't have, all of the, well, we'll talk about that in a little bit, but it doesn't have all of the material that it needs and machinery that it needs in order to swim or flagellate, right? By the time it's finished, meiosis 2, however, it begins to really become entry to spermatogenesis, which is becoming this like final like sperm itself. Okay. All right, so we'll talk a little bit about this in a minute, or like however long it takes you to watch the other video, I guess. And for now, I hope that was super helpful. I hope that gets you started, and I hope you go back and review meiosis and mitosis. And I hope you're having a great day. Bye-bye.